Okay, so good morning once again. Good afternoon once again. So for today, we will be talking about the microbial diversity of the eukaryotic microbes, or those organisms with true nucleus. Okay, so in relation to that, let us now we will be discussing the algae, the protozoans, the fungi, the lichens, and the slime mold. Okay, so let's talk about first the algae. Algae are considered to be as photosynthetic eukaryotic organisms. Now, always remember that whenever you hear the term photosynthetic in the food chain, they are always considered to be as the producer. So they are very important part of our food chain. Also, all algal cells consist of cytoplasm. So a cell wall, a cell membrane, a nucleus, plastids, ribosomes, mitochondria, and Golgi bodies. So just like your other these are the typical components of the eukaryotic cells. Now, some have a pellicle, stigma, and flagella. Some of them would have pellicle, stigma, and flagella. Algae ranges in size from being a unicellular microorganisms to large multicellular organisms. So such as the one that you can find in the sea, seaweeds or kelp. Yeah. So in fact, seaweeds are important source of agar. An agar or agar is the one that can solidify culture medium. Okay, so algae produce energy by photosynthesis. So some may use organic nutrient. Remember the term organotropes, organisms that will utilize organic nutrients. Okay, so algae may be arranged in colonies or strands and are found in fresh and salt water, in wet soil, or in wet rocks. So siguro you're familiar with lumot. Okay, so that's the common way of referring to algae in Tagalog or in Filipino. So we mostly call them lumot. Okay, so most algal cell wall contains cellulose. So cellulose makes it starchy. Okay, so that is the reason why we are actually using this. We are actually using seaweeds, for example, as the base component of agar. Okay, depending on their photosynthetic pigments, algae are classified as green, golden brown, or the red algae. So algae includes diatoms, dinoflagellates, desmids such as Spirogyra, Chlamydomonas, Volvox, and Euglena. Okay, so these are examples of algae. So algae are important source of food, iodine, fertilizers, emulsifiers, stabilizers, gelling agents for jam, and culture media. So if you're familiar with jelly or jam, do you know how to prepare them? We usually prepare them by boiling, and then they are being solidified when they are being placed in, if they are placed in a much lower temperature. And that's also how agar work in the culture media. Okay, so these are the morphologic characteristic of diatoms. Okay, so we also have here, so now we are going to discuss also protozoans, but first let us find that B and E, Euglena and Volvox. So this is Euglena, and this is Volvox. So these two are examples of algae, while the others are considered as protozoans. Okay, so algae are found in different environments, even in wet soil. Okay, so is algae medically significant? The answer is yes. In fact, one genus of algae the Protozoa is a very rare cause of human infections known as protothecosis. Okay. Algae in several genera would also secrete toxic substances called phycotoxins. So these are the red algae. So that's the reason why if you've been hearing the news red tide, so you do not want to eat shellfish if there is already red tide, because that particular shellfish, it would be producing a toxic substance known as phycotoxins. And phycotoxins are poisonous not just to humans, but to fish and other animals as well. So if we will be ingesting these phycotoxins, okay, they are produced by dinoflagellates that cause red tide. So it can lead to a disease called paralytic shellfish poisoning. So during red tide season, so you do not want to eat tahong, okay, you do not want to eat mussels, you don't want to eat, you do not want to eat oyster because of the possibility of the red tide. Okay? So this is the Protozoa cells, the causative agent of protothecosis. Okay. Now we're done with algae. Let's talk about protozoans. Okay. Protozoans belong to kingdom Protista. And there are three types of protists. We have the protophyta. We have the protophyta. And then we have the protomycota. And then we have the protozoa. Okay? Protophyta is the plant-like protist. Protomycota is the fungi-like protist. While protozoa, the one that we will be discussing today, this is the animal-like protist. Okay? So it is the animal-like protist. Okay? So protozoans are different from algae. Okay? Because they are non-photosynthetic. Okay? And they are eukaryotic organisms. Majority of the protozoans are considered to be as unicellular. So when you say unicellular, it means that these are single-celled organisms. So each cell is considered to be as one organism. Okay? And they are found free living and found in soil and water. All protozoal cells possess a variety of eukaryotic structures and organelles. Since protozoans do not have chlorophyll, they do not have pigment, they cannot make their own food by photosynthesis, unlike the algae. Therefore, they would need to ingest algae, yeast, bacteria, and even smaller protozoan as the source of nutrients. Okay, so they would need to ingest that as a source of nutrients. Okay, so protozoans do not have cell wall, okay, unlike algae, but some possess a thickened cell membrane called pellicle. And pellicle and cell wall would have the same purpose, and that is for protection. Okay, so protozoa would have a typical life cycle. The life cycle of the protozoan would have a typical two stages. So we have the trophozoite and the cyst. Okay, so what's the difference between a trophozoite and the cyst? The trophozoite is motile, feeding, and dividing stage. In fact, the most common way to describe a trophozoite is that we consider trophozoite as the feeding stage. Okay? The cyst, on the other hand, is non-motile, dormant, and survival stage. So survival stage, why? Because it is made up of a resistant, okay? They have a resistant pellicle, a more resistant pellicle. And that is the reason why we consider the cyst as the infective stage. So most of the time, we consider the cyst as the infective stage of the parasite. Okay? Some protozoans are parasites. What do you mean by the word parasites? Okay? Parasites are organisms that cannot survive without a host. Okay? There are several kinds of parasites, and protozoans is just one of the parasites. And examples of these parasites are malaria, which is caused by the Plasmodium species, Giardiasis. Giardiasis is caused by Giardia lamblia, and trypanosomiasis. Trypanosomiasis is caused by Trypanosoma cruzi. Okay, so there are many kinds of Trypanosoma species. So I'm just mentioning the most common ones. Okay, so these are examples of parasitic protozoans. Protozoans are sometimes divided into groups based on their locomotory organelles. So we have the amoebae, we have the ciliates, we have the flagellates, and then we have these sporozoans. So when you say amoebae, the main locomotory organelle is called pseudopodia. Pseudopodia is really not a locomotory organelle. In fact, the term pseudo means false, while podia means feet. So the term pseudopodia means false feet, because these are not really foot, but the amoeboid movement, the so-called amoeboid movement, is just like a cytoplasmic extension. Okay, so amoeboid movement are just like cytoplasmic extension. Ciliates, on the other hand, are actually caused by is actually look like small hair-like structure. So this is how cilia look like, and the most common protozoan with cilia is called Balantidium coli. The causative agent of balantidiasis, which is Balantidium coli, by the way, is the largest intestinal protozoan infecting humans. Okay, the number 3 types are the flagellates. Flagellates move by means of flagella. So Giardia lamblia, the causative agent of giardiasis, will cause flagellates. And sporozoans do not have means for locomotion. They do not have visible means for locomotion. So sometimes it is referred as protozoans with no specific locomotory organelles. So that's how they describe sporozoans. So this is an example of amoeba, and this is the pseudopodia. So pseudopodia are not really foot. Okay, they are called false foot. So it merely moves by means of cytoplasmic extension. So this is Balantidium coli, and this is how cilia look like. So small hair-like projection. And this is the Giardia lamblia, and these are the different flagella of Giardia lamblia. Sorry, my slides are not moving again. So my apologies for that. Okay, so let me share my presentation again. And it's not visible. Ayan. So this is the pseudopodia. So this is an example of amoeba. Okay, this is an example of amoeba, and as you can see, this is the pseudopodia or the false foot. So these are not really foot, but this is not really foot, but it merely moves by means of cytoplasmic extension. And then this is the cilia of Balantidium coli, the small hair-like projection surrounding the entire cell. And these are the flagella of Giardia labria. Okay, so let's talk about fungi. Okay, the study of fungi is called mycology, and scientists who studied fungi are called mycologists. So do you know that fungi are virtually found everywhere? They are ubiquitous. Some of them are harmful. So if they are harmful, they can cause fungal diseases. And fungal diseases is termed mycosis. Okay, some are beneficial. So fungi represent a diverse group of eukaryotic organisms, including the yeast, molds, and fleshy fungi, which sometimes you could eat, such as mushroom. Okay, fungi are the so-called garbage disposers of nature because we consider them as saprophytes. So when you say saprophytes, they live on decaying organic matter. So they are decomposers, they are saprophytes. Fungi are not plants. Therefore, they do not have chlorophyll and they are not photosynthetic. Okay? Fungal cell walls contain a polysaccharide called chitin. So this is the main component. So remember, if bacteria have peptidoglycan, fungi would have chitin. Okay? Some fungi are unicellular. So an example of unicellular fungi are yeast. Okay? Yeast such as Saccharomyces cerevisiae, the one that we're using for fermentation of wine. Okay? While other will grow as filament called hyphae. So this is an example. The one that I'm drawing here is a hyphae. If hyphae intertwine with one another, it is now called mycelium. Singly, it's called hyphae. But if it intertwines, then it is now called a mycelium. Some fungi would have a septate hyphae. So for example, this is how a septate hyphae would look like because there are cross walls. Okay, or septa. Some fungi would have a septate hyphae, which means that it's all continuous. Okay, whether or not a fungus has a septate or septate hyphae, it is important clue to identify certain types of fungi. Okay, so depending on the species, fungal cells can reproduce by means of budding. So usually budding is for yeast, hyphal extension, or the formation of spores. So fungi would have spores, but remember, bacterial endospores are different from fungal spores. Bacterial endospores are mainly used for survival, but the fungal spores are mainly used for reproduction. And there are two types of sexual spore of the fungal spores. No, we have the sexual spores, and the other one are the asexual spores, otherwise known as conidia. So some fungi will produce both asexual and asexual spores. And fungal spores are very resistant structure. Now, there are some fungi that do not produce sexual spores, and we call them fungi imperfect. Fungi without sex life or without sexual life cycle. Yeah. So again, let me show you how fungi look like. No, this is a single hyphae that has cross wall. So that's the reason why we call it septated hyphae. So if no cross wall, it is just continuous, then it is called a septate hyphae. If the hyphae intertwine, we call it mycelium. Okay? And if this is the culture medium, and if the hyphae is found at the surface, we call it the aerial hyphae at the surface of agar, and surface aerial hyphae is important for reproduction, while those that are submerged in the agar is called vegetative hyphae, and its main function is for absorption of nutrients. So it appears like roots, okay, for absorption of nutrients. But these are not roots because roots are supposed to be found in plants. So instead of calling it roots, we call it vegetative hyphae. So how do we classify fungi? So fungi is based on their mode of, are classified based on their mode of sexual reproduction and the type of sexual spore they produce. So because of that, we can actually categorize fungi into 5 phyla, namely the Zygomycotina, the Chytridiomycotina, the Ascomycotina, the Basidiomycotina, and then the Deuteromycotina. So Deuteromycotina or the Deuteromycetes includes medically important molds such as Aspergillus and Penicillium. So fungi in this phylum have no mode of sexual reproduction, walang sex life, or the mode of sexual reproduction is unknown. So this is the one that I've been telling you a while ago. So this is the fungi imperfecti because of there's no sexual reproduction. So these are examples of fungi. So the different kinds of fungi as shown under the microscope. Okay, so we have the sporangiospores. So under the sporangiospores, Rhizopus is a species of Zygomycetes. So here, the entire sporangium contains different kinds of spores. And then we have the Aspergillus species, and the Aspergillus species has conidia. Okay, and the conidiophore contains the conidia. Okay, let's talk about yeast. So yeast is a type of fungi, and yeast are said to be eukaryotic unicellular organisms. Unlike the multicellular fungi, they do not form mycelia. So individual yeast cells are considered as an organisms already. So that's why they are unicellular. So they are also known as the blastospores or blastoconidia, and it can only be seen if you will be using a microscope. So yeast reproduce by means of budding. So one yeast will produce a daughter cell. So this is what you call budding. Okay? And a string of elongated buds is known as the pseudohyphae. So some yeasts are capable of producing a pseudohyphae when there are strings that elongates from the buds. So some yeasts produce thick walled spore-like structures called chlamydospores or chlamydoconidia. So this is an example of a yeast. So this is the mother cells. The mother cell buds into a daughter cell. Okay, so this is the budding yeast. Okay, so letter A is an example of chlamydospore. So this one is a thickened cell wall. Letter B is the pseudohyphae. These are not real hyphae, but these are just elongated strings that attach from the bud. And letter C, the one that you can see here, is the budding yeast that can produce the blastospores. Okay, so this is Candida albicans, another example of yeast. And you know, Candida albicans is a common opportunistic infection in HIV, and they can also cause thrush. It's an oral mucocutaneous candidiasis. Yeast are found in soil and water and on the skins of many fruits and vegetables. So yeast have been used for centuries to make wines and beer. So like what I told you, Saccharomyces cerevisiae is the yeast that we are using in baking, yung pangpaalsa. Okay? Candida albicans is the yeast most frequently isolated from human clinical specimen and is also the fungus most frequently isolated from human clinical specimen, particularly if a person is immunocompromised because of the HIV infection. So yeast colonies may be difficult to distinguish from bacterial colonies because both of them are creamy. Therefore, in order for you to differentiate it from bacteria, so you can simply perform a wet mount. So you place the yeast, the smear on the slide, okay? So yeast are larger than bacteria and are usually oval shaped, okay? Yeast are often observed in the process of budding. So bacteria do not exhibit budding. So that's how we differentiate yeast from bacteria. So again, this is an example of a yeast. Okay, this is an example of bacteria. These are WBCs. So that's how they look like under the microscope. So this is the colonies of Candida albicans on blood agar. So you can actually take note that they are somewhat similar to the colonies of bacteria. Now let's talk about molds. So sometimes it is spelled as molds, or so I think this is the British spelling, no? But nonetheless they're the same. So molds are often seen in water and soil growing on food. So in Tagalog we call it amang. Okay? So molds produce cytoplasmic filaments called hyphae. And I think I have already discussed to you the difference between the aerial hyphae and vegetative hyphae. So aerial hyphae grows above the surface of whatever the mold is growing on. So usually it is being used for reproduction. Okay. On the other hand, vegetative hyphae grows beneath the surface. So most likely for absorption. So reproduction is by spore formation, either sexually or asexually, on aerial hyphae. So that's the reason why the aerial hyphae is sometimes known as the reproductive hyphae. So molds have great commercial importance. Do you know the first antibiotics was discovered by Alexander Fleming from a mold known as Penicillium notatum. So this is the first antibiotics, and from Penicillium notatum, they were able to discover the active component, which is the penicillin. Okay, so the flavor of blue cheese, Roquefort, Camembert, and Limburger, these are all expensive cheeses. Okay, the flavor are actually due to molds that grow in them. So if you like blue cheese and you like the taste, so you are actually liking the taste of the mold. Okay, so these are expensive cheeses. So this is the mold on bread. So pag nakakita kayo ng ganito sa tinapay, do not eat them anymore. Okay. So let's talk about the fleshy fungi. So these fungi include the mushrooms, toadstools, puffballs, and bracket fungi. So they consist of network of filaments of strands. So we call that mycelium, and they can grow in soil or even on a rotting piece of wood. So the fruiting body that grows above the ground will form and will eventually release the spores. And that's the reason why you can see umbrella-shaped like structure. So we call that the fruiting body. So some mushrooms are edible. In fact, mushrooms are my favorite, but do not just eat mushroom because some are extremely toxic. And the toxins that are being produced by mushrooms, we call it the mycotoxins. So this example of fleshy fungi growing on growing on the forest floor. Okay, so let's talk about medically important fungi. So a variety of fungi, including yeast, molds, and some fleshy fungi are medically important, or they're also important in the field of veterinary medicine and also in agriculture because they can cause diseases in humans, animals, and plants. But of course, our concerns are humans. So if they can cause infections to us, so the type of infection is called mycosis. And there are actually 5 categories of mycosis. We could have the superficial mycosis, okay, cutaneous mycosis, subcutaneous mycosis, systemic mycosis, and the 5th one is called opportunistic mycosis. Okay. Superficial mycosis are fungal infections of the outermost area of the human body, such as the hair, nails, and epidermis. So an example of that is malassezia furfur. Malassezia furfur. Okay? So causes, in Tagalog, we call it an. So that's an example of superficial mycosis. Okay? And then cutaneous mycosis are fungal infections of living layers of the skin and the dermis. So a group of molds collectively known as dermatophytes causes tinea or the ringworm infection. Now it's a misnomer because they thought at first that fungi are plants. So they call it dermatophytes. Phytes means plants. And it's like a ring-like structure. They thought that it was a worm. So they call it a worm. But these are not worm. These are not plants. That's the reason why the better way to describe this one is dermatomycosis. Okay? So it says here, note that ringworm infections have nothing to do with worms because they are not caused by worms. So the yeast Candida albicans can also cause cutaneous, oral, and vaginal infections. Okay? So subcutaneous mycosis and systemic mycosis are most severe types of fungal infections. So subcutaneous mycosis will now infect area beneath the dermis and underlying tissues. So this would already include muscles. Okay, so Madura foot is an example of subcutaneous mycosis. Okay, so then systemic mycosis are fungal infections of the internal organs of the body. So majority, we can actually get systemic mycosis by inhaling the spores of these fungi. So spores of some pathogenic fungi may be inhaled with dust from contaminated soil or dry bird or bat feces. They may also enter through wounds of the hands and feet. So when we inhale the spores, then we can get systemic mycosis. So example of deep-seated pulmonary infections include plastomycosis, coccidioidomycosis, cryptococcosis, and histoplasmosis. So these are all examples of systemic mycosis. And since this pulmonary, you usually get it through inhalation of spores. There are some mycoses that you can inhale from bed molds, such as Rhizopus and Mucor. These are okay for us, for those of us who are perfectly normal in terms of the stability of our immune system. However, if a person is immunocompromised and was able to inhale bread molds, that would be deadly for them. Because this bread mold could become what? An opportunistic mycosis. It could become opportunistic mycosis. So diagnosis of mycosis is accomplished by culture techniques and immunodiagnostic procedures. So yeast are identified using a series of biochemical tests, and molds are also identified by the rate of growth and by using a combination of macroscopic and microscopic observations. So usually mycologists would have the expertise to identify these molds. Now let's talk about dimorphic fungi. Okay, so the term dimorphic means two forms or two morphology. And dimorphic fungi is temperature dependent. So dimorphic fungi are temperature dependent. So a few fungi include some pathogens that can live as either yeast or mold depending on the growth conditions. So we call it dimorphism. Okay? So I said it's temperature dependent because some fungi, when they are inside our body and the temperature is 37 degrees, they become yeast. However, when they go out of our body and the temperature is at room temperature, they become a mold. Okay? So remember in dimorphism, at 37 degrees, they are yeast. At 25 degrees, they are molds. Okay? So this is what you call dimorphism. Okay? So in vivo, dimorphic fungi, when you say in vivo, inside our body, so they exist as yeast. But in vitro, when you say in vitro, which means outside our body, they exist as molds. Okay, so these are dimorphic fungi. So these are examples of dimorphic fungi. So at 37 degrees, they are yeast. At 25 degrees, they are molds. Okay, and first, let's talk about, lastly, the lichens and the slime molds. Okay, so lichens are observed as colored, most of the time circular patches on trees and tree trunks and rocks. So in a tree, pag meron kayo napapansin na circular patches na whitish, yan, it would be a lichens. Do you know that lichens are composed of an algae and a fungus living in mutualistic relationship, which means that algae benefits from fungus and fungus benefits from algae. So lichens are classified as protists. Slime molds are another different organisms. No, slime molds are found in soil and on rotting logs. So slime molds have both fungal and protozoan characteristics. So slime molds are classified also as protists. Okay, so lichens are protophyta, slime molds are protomycota. So again, protophyta, plant-like protists, slime molds are fungi-like protists or protomycota. Yan. So these are example of slime of lichens. So you can see the circular patches. Okay. So slime molds naman on the forest floor. Okay, so that ends our discussion on eukaryotic organisms. Okay, so thank you very much for bearing with me.